Welcome back, guys. Let's talk about fertilization today. By definition, fertilization is a sequence of biological processes initiated by entry of a spermatozoon into a mature oocyte, followed by formation of pronuclei. What is a site for fertilization? It is the ampullary part of fallopian tube. Fertilizable lifespan of human oocyte ranges between 12 to 24 hours. Events of fertilization. First is the preparation and transport of spermatozoon and ovum, followed by capacitation and contact, acrosome reaction and penetration, fusion of the sperm with the egg, followed by activation of the ovum. So we will discuss all these one by one. Let's talk about the preparation of sperm. The sperm takes approximately 72 days to reach cauda epididymis after initiation of spermatogenesis. At this stage, the head of the sperm contains a membrane-bound nucleus capped by acrosome. Acrosome is a large vacuole of proteolytic enzymes. Motility and ability to fertilize are attained gradually as the sperm pass into epididymis. Cauda epididymis stores the sperm available for ejaculation. Preservation of optimal sperm function requires adequate testosterone levels as well as a normal scrotal temperature. The ability to store functional sperm provides a capacity for a male to have a repetitive fertile ejaculations. Sperm acquires surface proteins in the epididymis that are later removed during capacitation. Semen forms a gel almost immediately after ejaculation, but then is liquefied after 15 to 20 minutes due to prostatic secretions. The alkaline pH of the semen provides protection from the acidic environment of the vagina, but this protection is very transient, and most of the sperms are immobilized within few hours. Contractions of female reproductive tract during coitus may be important for entry of the sperm into cervical mucus and further transport. The sperms swim and migrate through the pores in the cervical mucus that are smaller than the sperm head. Therefore, the sperms must actively push their way through the mucus. Beta defensin 126 is acquired in the cauda epididymis, which carries a high negative charge and is critical for movement through the cervical mucus. We can see here the attrition of sperm number from vagina to the tube. Ejaculated sperm show a drastic attrition while they enter in the vagina and further ascends the cervical mucus, ultimately reaching up to the fallopian tube. Preparation of oocyte. The fimbriated end of the tube sweeps over the ovary to pick up the egg, and its entry into the tube is facilitated by muscular movements. We have discussed all these things in the lecture on oocyte transport, and we already discussed that how the fallopian tube plays an active role in sweeping the egg inside the fallopian tube. The cilia on the fimbrial surface display adhesive sites, and they seem to be responsible for initial movement of egg into the tube. Capacitation has already been discussed in the previous lectures. Just a recap: the cellular changes that ejaculated spermatozoa must undergo in order to fertilize is called capacitation. It is initiated during sperm's passage through the cervix and is completed in the fallopian tubes, and is characterized by three accomplishments: ability to undergo zona reaction, ability to bind to zona pellucida, and acquisition of hypermotility. Acrosome reaction and penetration. When the acrosome reaction occurs, a number of proteolytic enzymes are exposed or released. One or more of these enzymes is responsible for digesting the whole through the zona pellucida, through which the sperm enters the perivitelline space. It requires influx of calcium ions, efflux of hydrogen ions, increase in the pH, fusion of plasma membrane with the outer acrosomal membrane. In freshly ovulated eggs, the ZP3 is very close physical association with ZP2. Acrosome reacted sperm bind to ZP2 via their exposed inner acrosomal membrane, penetrate the zona, fuse with the oocyte, and fertilize the egg. Immediately after fertilization, the cortical granules release a ZP2 specific protease into the perivitelline space. The ZP2 specific protease clips ZP2 and converts it into cleaved form, that is ZP2C, that no longer support the binding of acrosome reacted sperm. We can see here in the first one, the sperm is making contact with the egg. Then acrosome reaction is happening with zona pellucida. The third one, acrosome reacts with perivitelline space, the yellow one. The fourth one, the plasma membrane of sperm and egg fuse. This is the plasma membrane of the egg, and sperm is fusing with it. The sperm nucleus is entering into the egg. This is the sperm nucleus. And finally, the cortical granules fuse with the egg plasma membrane, which renders the vitelline layer impenetrable to sperm. Sperm oolyma binding. The fusion of sperm with oolyma causes weak depolarization, resulting in a calcium wave. Increase in calcium signals the oocyte to resume cell division, thus completing the meiosis 2. Intracellular calcium triggers the fusion of cortical granules with the oolyma. We can see here in this image, the acrosome reaction is happening and the sperm egg fusion is happening. That is leading to release of the phospholipase C zeta, which helps in the calcium release and that leads to calcium oscillations. There is something known as cortical reaction. It is exocytosis of lysosome-like organelles from the cortical granules. Zona reaction is as soon as these organelles are released in perivitelline space, they interact with zona pellucida. Due to release of hydrolytic enzymes in cortical reaction, the zona hardens by cross-linking of proteins and inactivation of ligands for sperm receptors. Thus, zona block to prevent polyspermy is accomplished. So thank you guys. This was all about the fertilization, and I know these topics are a little off the track, and they are from the because they are the basics of reproductive medicine. But as long as we will not clear our basics, the moving directly to the clinical aspect is not a good sign of a good student. So we must read the basics first, and then finally moving on with the clinical aspects is the best thing what a student can do. And that's why I'm putting so much emphasis on clearing the basis. Please write in the comments if you want any other topics need to be covered. Thank you.